Hello, my name is Pat. I'm from the MIT Media Lab, and today I would like to present our paper, Living Bit: Opportunities and Challenges for Integrating Living Microorganisms in Human-Computer Interaction. Before we begin, just a word of caution. This work is unconventional for augmented humans community as we attempt to think beyond digital computing and traditional boundaries that exist between biological cells and computers in order to integrate living bits or microorganisms for human augmentation. With ubiquitous nature of computing, the relationship between humans and computer is becoming more intimate. Each of the era of computing is characterized by the unique ratio between users and machines, as shown in the slide here. In addition of having computers closer to the human body, there are trillions of living computers on, inside, and around us, microbes. The term microbes or microorganism refer to micron-scale living organisms, including bacteria, yeast, and fungi. The reason we call microbes living computers because in principle, biological cells compute to build. A living cell could receive input, such as chemical cue or physical signals, from its surrounding to compute, process, and metabolize in order to respond to the environment. The microbe is used as one of the model organisms for studying and engineering biological computation and fabrication in modern biotechnology research. For example, biopharmaceutical companies have successfully inserted a human gene responsible for producing insulin in E. coli cell to help individuals with type 1 diabetes producing insulin. In the past decade, researchers have used the ability of microorganisms to produce output such as chemicals, smell, taste, color, movement, and more as part of interactive systems ranging from wearable device to musical instrument, built environment, food, games, robots, and demonstrating a rich spectrum of research in HCI that engaging with microorganisms. These microorganisms have been integrated with human life for thousands of years. We have used them in form of ancient technology to create, transform, and preserve materials and chemicals such as food and agricultural products. As Mark Weiser, pioneer in ubiquitous computing, said, "The most profound technologies are those that disappear. They weave themselves into the fabric of everyday life until they are indistinguishable from it." His philosophy also applied to biotechnology, one of the oldest and most impactful technologies that humans ever invented. In this paper, we introduced Living Bit, the integration of microorganisms in, with, and as computing system. Inspired by how tangible bits sought to bridge the gap between digital and physical environment, and how Parks and Dickey create a biological imperative for design interaction, Living Bit is an attempt to think beyond the traditional boundaries that exist between biological cells and computers. Our goal for Living Bits are to, first, outline and inspire the possibility of HCI researchers to rethink computation between digital technology and organic and regenerative living systems such as microorganisms. Second, explore and characterize novel human-microbe interactions across context and scale. Third, provide principles for stimulating discussion, presentation, and brainstorm for future microbial and biological interface design. We aim to make Living Bits accessible for researchers across SCI, synthetic biology, biotechnology, and interaction design to explore the next generation of biological human-computer interaction. We conduct a search across multiple existing fields and select example projects that are well established in each communities, have been published and exhibited to the public. These selected examples represent the rich spectrum of research within microbial HCI. The selected microbial interfaces project include what is shown here. We use the example as the starting point for investigation. In the process for conceptualizing Living Bits, we characterize and systematically study the utilization of microbes as biological interface in each project across scale and applications. We present our analysis in three parts. First, the parallel between microbial computation and digital computation. Second, the different application domains. And third, the design element of microbial interface. Due to time constraint, we will discuss the highlight for each section. The parallel between microbial computation and digital computation. Living cells share many characteristics with a digital computer, in the sense that both systems can receive input, information, compute, and then respond. However, both have unique advantage and disadvantage. In terms of input, process, and output, microbes are flexible compared to digital computers. Input to the microbial cell can be molecular, which include chemical, ionic, nucleic, and antigen molecules. Input can also be physical conditions of the environment such as light, heat, magnetic fields. Similar to the input channel, microbes have wide range of output modalities. Output can be visual, olfactory, mechanical, and chemical. Examples include producing pigment or fluorescent molecule for visual cues. In digital computing, logic gates have binary 0 or 1 output in response to one or more inputs. In synthetic biology, cells can simulate this behavior when these biological logic gates are combined. It is possible to simulate digital-like computation solely using biological function. Researchers have demonstrated the ability to toggle between biological functions in binary fashion through the genetic toggle switch in bacteria. Researchers have also developed complex genetic circuits with multiple logic gates, as well as designing programming language and software to create them. For example, Autodesk developed Genetic Constructor, a cloud-based computer-aided design system to support the design of genetic circuits using visual language that focus on the functional part of the gene. The genetic circuit allows researchers to sense, compute, and actuate in the microorganism. The rest of analysis in terms of programming language, scale, speed, power source, and recyclability can be found in the paper. Different application domains. We identify 5 different categories of how microbes have been used as interface in the context of HCI to embody, communicate, enhance, materialize, and play. Embody microbial interface are those that capture and translate digital and experimental data into something tangible. Embodiment is defined as a physical manifestation of computational artifact or phenomenon. In Vesper, the researcher conceptualized the process of embedding engineered microorganisms in the 3D printed dead mass that could embody the last breath of a human being by converting chemical component of the breath into visible pigment, transitioning from vessel of representative neurovasculature to actual biological urns. The researcher further characterized such composite material as hybrid living materials. In Biotabit, researchers explored the concept of musical embodiment by designing a microbiome record player. The player used an algorithm to extract location, density, and cluster size from petri dish that contain microbe culture from different parts of the human body to algorithms that generate musical composition. The microbiome record player is currently used as an outreach tool for public to learn about music of their microbiomes. Microbes have been used as a medium for communication through different signals across scale and environment. They work as signal amplifier by taking the input and turning it into observable and measurable output. In Ecromy project, researchers used microbes to develop a yogurt which changed an individual feces color in response to specific health and disease biomarkers. They accomplished this by engineering pigment production via metabolic pathway inside bacteria. By connecting the pathway for producing visual output with sensing modality of the cell, researchers can integrate living organisms as ubiquitous sensors that communicate the surrounding information. For example, researchers have demonstrated the use of stretchable, robust, and biocompatible hydrogel to host engineered bacteria to create chemical sensors that can be attached to the body and robot. Biofabrication is one of the largest domains of research in biotechnology and biodesign. Several types of microbes such as bacteria and fungus naturally produce biomaterial as the byproduct of their metabolism. Researchers have developed processes for cultivating microbes to produce biodegradable materials such as cellulose, mycelium, and others. These materials have been woven into biological interface across application. For example, artists and designers have created wearable microbial 3D fiber art and a fashion collection BioCouture using microbial cellulose produced from kombucha culture of yeast and bacteria. The research team proposed the application of microbial cellulose as biological exoskeleton of electrical system with renewable, self-healing, and shape-changing properties. Moreover, researchers proposed using biodegradable materials to create more sustainable electronic life cycle. Enhance. Through embedding living organisms inside everyday objects, researchers could tap into biological functions found in microbes to augment capabilities of materials and devices. This enhancement, ranging from photosynthesis to shape-changing ability, could lead to symbiotic relationship between human and microbes. For example, Mushtari is a wearable embedded with synthetic microorganism that can augment human biological functionality for space travel. Researchers 3D print a wearable microfluidic channel containing 2 genetically engineered microorganisms, a photosynthetic type such as microalgae or cyanobacteria that can convert sunlight into sugar, and compatible microbes such as yeast and E. coli that convert sugar into functional materials for wearers such as drug, food, fuel, and more. Play. Researchers introduced the concept of a biotic game, a game that incorporates a biological system as gaming element. The biotic game is created to motivate students learning at the intersection of life science and device engineering. The game used the real-time interactivity of living microorganisms, turning classical observatory microscope into an interactive experience. In Euglena soccer game, the player needs to score point by observing and controlling the position of Euglena to shoot virtual ball into the goal. Euglena is a microorganism with a whip-like appendages called flagella used to move around. The game developer used photostaxis, a behavioral microorganism that move toward light. Thus, player orient the Euglena using 4 LED, one in each corner. Morash is a hybrid biotic game that is played on a slow but constantly evolving biological landscape of living microorganisms such as bacteria, yeast, and fungi. As different species grow and move across nutrient terrain, they can be digitally captured with high-resolution scanner for the player to observe any physical change when they occur. The goal of Morash is to compete with the fellow players and collect as many microbial cells as possible over a 5-day period. Points are awarded based on the coverage of the cell. The research team discovered that the slowness of microbial growth may not necessarily compromise playing experience, but rather enhance it, showing a promise for future interactive biotechnology. Finally, design element of living bit. The project presented in the previous section demonstrates how SCI researchers have already begun paving the way to biological interface by exploring how to work and design with microbes. This body of research is growing and there are many more possible future directions. However, to systematically understand the biological system, we describe living bits in 5 levels: cell, colony, system, interface, and interaction. We arrange these levels from smallest to largest scale. The use of microbes as computer is not limited to scientists, researchers in academia or industry. DIY or do-it-yourself and citizen biotechnology communities have begun making progress toward democratizing biotechnology and synthetic biology source code. As citizen science community continue to grow, microbes have become the key material entry point to the world of biotechnology. Many open source protocols on how to isolate, cultivate, and engineer microbes using everyday materials and tools are becoming more available online. Moving forward, applying the concept of living bits to design research could be an intimidating process, as every paradigm shift requires courage, a spirit of experimentation and exploration. To mitigate this process, we come up with 4 steps to help researchers integrate microbe into SCI projects. Living bits offer the opportunity to harness natural properties of microorganisms either in or beyond traditional computing paradigm and architecture. It can have positive impact on our digital life, health, and natural environment. However, technical and societal and ethical challenges remain. We aim to stimulate discussion and debate about the future of microbial interface, even biological SCI at large, and its impact on society. We hope to encourage SCI researchers to approach non-living and living material as continuum of computational interaction. Living bit raised the ethical concern regarding the integration of biological matter as design elements. We know synthetic biology has potential to affect all peoples, positively and negatively, yet we do not have a complete understanding of biology. Further, manipulating the genetic materials and behaviors of the microbes in the design process may contribute to anthropocentrism, a problematic point of view that humans are the only important entity in the ecosystem. In this paper, we study microbial interface in the context of SCI, which may support this notion. However, living bits is the idea of coexistent computation. Therefore, researchers that work with microbial interface should think of human-microbe interaction as a symbiotic relationship rather than the materialistic exploitation of biological entities. In response to the rapid evolving body of biotechnology research, New Direction, the Ethics of Synthetic Biology and Emerging Technologies, compile views of science, ethics, and social issues of synthetic biology. The book was published by the US Presidential Commission for Study of Bioethics. Though it's focused on scientific research, it can be extended to microbial SCI and living bits as well. In conclusion, microorganisms form the foundation for human life. Their existence predates ours by 3 billion years and is crucial to the basic functioning of our brains and body. In addition, we have adopted microbes as a technology. We have worked with microbes to ferment food and alcohol for thousands of years, all the way to genetically engineer modifying microbes to cure disease today. Recent advancements in synthetic biology have led to the ability to adopt microbes as computers. These tiny organisms can be programmed to accept a wide variety of input and subsequently exist in binary states, akin to logic gates that form basis of computing. But these organisms have an incredibly wide range of capability compared to traditional circuits. They can self-heal by growing and multiplying without outside interference. They can create renewable materials and energy and even turn specific color to report human health and disease condition. All of this in a package 1,000 of a meter or smaller. Living Bits aim to bridge the digital and microbial entity. We show how researchers can integrate microorganisms into technology projects to the parallel between digital computing and biological computing, different application domains, and scale of living bits. We aim to help researchers understand microbial HCI and its opportunities and challenges. We believe that as we integrate biology and computing together, we can shape the new era of HCI beyond digital toward organic computation to regenerative living system. Thank you.